All right, let me record. Okay, so let me go back to that. Yeah, I think why is it so slow? So when it opens, I'll just show you real quick. But again, you're just gonna listen to my video, have the PDF ready as you're listening. And then, like I said, on Thursday, I may then just show you guys the graphs a little bit more if you guys have questions about it, okay? But I want you to be able to get started on this if you want to, okay? So again, you're not gonna be handing in this PDF, right? Completed with Cami or anything like that. You just need this with you, one, to read the background information, but then also to be following along what I'm doing. So I'm basically gonna be doing these experiments, and then you guys are gonna be recording that data, okay? But again, the assignment is gonna be a lab report, just like you guys did for the last couple labs, okay? So let me just go back. Okay, so this is the actual assignment. You see there's a sample lab report there. I'll show you that again on Thursday, just because it's better if some of you guys have already kind of attempted to do the lab or have done the lab. If I show that to you now, it's not gonna mean anything. So again, here, right? The instructions for the lab report are there. Make sure you take a look at it. Like I said, what's different about this lab report is that there's some graphs that you need to include, okay? And again, I do explain that in the video, but in terms of making a graph in Excel, if you need a little bit more information on that, I can go through that with you guys in a little more detail on Friday, okay? Or on Thursday. Oh my goodness, on Thursday. All right, guys, so is that clear? So this Enzyme Lab PowerPoint is just some sort of introductory information that I go through again in the video, right? So basically in the video, I'm gonna tell you exactly what you guys need to be looking at and explain everything there. All right, does anyone have any questions about that? No? Okay, so like I said, this will be due next week. I'll probably make it due that Friday, next Friday, okay? But you'll definitely have a week to do it. Okay, so that's what I was gonna do during the lab Zoom meeting. Don't worry about logging in. Like I said, for the graphs that you're gonna make in Excel, I'll probably revisit that again on Thursday during class, okay? Just to make sure everyone knows what to do for that. So it would be to everyone's benefit to at least take a look at this and kind of start it so that you understand what I mean when I'm talking about the graphs. Okay, guys? Because if I talked about the graphs right now, you're gonna be like, what the heck is she talking about? So it would be good if you guys could try at least get some of it done or listen in on some of it before class on Thursday. Okay.
Professor?
Yeah, go ahead.
My audio cut out. Did you say that was what we were going to go over during the lab meeting?
We were going to go over that during the lab meeting, so I'm not. So we're not going to have a lab meeting. So you don't.
Okay, so we're not logging on for the lab?
Correct. You don't need to worry about logging on to Zoom for the lab meeting. I just figured it doesn't make sense for me to do it twice. Okay. But like I said, on Thursday, I will talk a little bit more about the graphs and the lab report if necessary. Okay. So everyone hopefully can, start this before Thursday. Okay. All right, so like I said, we're starting chapter 8. So let me just pull up chapter 8. I also remember I tried to show you guys some videos or animations from chapter 7, and it wasn't working in the room that I was in. So I just want to kind of just pull up maybe 1 or 2 from chapter 8 just to kind of help introduce that chapter. And then, like I said, we're going to kind of end with you guys basically working on something. On a Google Doc in groups, and then you guys can just share it with me and we'll sort of end class that way. So that's essentially what we are doing. So let me just talk about chapter 8, because again, maybe a lot of you guys haven't listened to the recorded lecture yet. So I'm not gonna go too much in depth with chapter 8, just kind of introduce it. And then we can talk more about it on Thursday as well. Okay, so what did I want to show you guys? Oh, 1 second. Actually, you know what? Let me pull up chapter 8 first. Okay, so chapter 7 was about photosynthesis, right? Chapter 8 focuses on cellular respiration. I told you that these 2 chapters are, they're a little rough, okay? And so they're pretty dense in terms of information, and it's like biochemistry sort of based, and so some of the concepts are a little more complex. Again, there is a figure here that I think is really helpful in terms of like big picture, okay? So Remember how I told you that one photosynthesis figure I think is really important because it's understanding the big picture, okay? So for cellular respiration, this is the figure that's important, or again, maybe not most important, but important to kind of really understand first before you start learning some of the details. So if you guys take a look here, there's basically 4 main phases. So it says here, 4 phases, go away, 4 phases in basically complete glucose breakdown, okay? So why does the cell, I'll ask you guys, right? Why does the cell break down glucose? What's the purpose of doing that? Somebody tell me, why does the cell break down glucose? Somebody? Why does the cell break down glucose?
For energy.
For energy, thank you. Okay, for energy, right? That's how the cell's gonna generate ATP. So whenever you talk about cellular respiration, which essentially is kind of just the general term, we're talking about the cell breaking down glucose to generate energy, okay? And there are 4 phases that are involved in this, right? The first one showing here, glycolysis, okay? So the reason why this kind of series of slides is super important is because, again, it puts everything kind of all together, okay? So a couple things, it's showing you where these things are happening, right? So we're inside the cell, glycolysis, where is glycolysis happening in the cell? So this is inside the cell here where we're at, right? Inside the cell. This is the mitochondria. So where's glycolysis occurring in the cell?
Is it the matrix in the mitochondria?
The cytoplasm?
Yeah, the cytoplasm. Thanks. Okay. So glycolysis happens in the cytoplasm, not in the mitochondria. Okay. And glycolysis is the first part of cellular respiration, right? Or if we're talking about breakdown of glucose, this is the first phase. Okay. So in glycolysis, right, you start with glucose and you end up with pyruvate, 2 molecules of pyruvate. Okay. In the process, if you notice here, right, you're going to generate on the bottom, it'll show you what ATP is generated, okay? So you get 2 net ATPs generated, but you also get NADH produced, okay? Where's this NADH going? Where's that red arrow going in this figure?
To the mitochondrial matrix?
Yeah, to the mitochondrial, well, essentially it's going to the inner membrane there, but to the electron transport chain, okay? It does look like it's going in the matrix the way the arrow is pointed, but it's going to the electron transport chain. So do you remember in photosynthesis when we talked about that electron transport chain, right? That's where ATP is generated. Remember we talked about the hydrogen ions that accumulate on one side, right? And then they move through a certain sort of pump or enzyme. Do you guys remember what the name of that is that actually generates the ATP?
ATP synthase?
Yeah, good, ATP synthase. Right, so this is the same idea here. All of these NADHs, you'll see as we move through the different phases that are produced, they go to the electron transport chain because they serve as electron, donors essentially, right? So electrons get passed, they serve as the source of electrons, okay? We'll go there for the electron transport chain. Once electrons are moved through that electron transport chain, then we can get ATP produced, okay? So sometimes you'll generate ATP directly from these processes, right? And then sometimes molecules like NADH will be produced, okay? Both are equally important in terms of generating energy, right? So if we move on to the next phase here, the next phase we'll call the preparatory reaction. So some books won't divide this up as a separate phase, but we will. So we're gonna call this the prep reaction, okay? And this is gonna occur in the mitochondria. What do you notice? There's more NADH and also FADH2 that's produced. Again, those go to the electron transport chain, ultimately is gonna help generate more ATP, okay? Next is citric acid cycle. Okay, citric acid cycle, you can see there's some net ATP gain, but then also NADH and FADH2 that's produced, okay? So sorry, from the prep reaction, it's only NADH that's produced. And then finally, if you go to the final phase, which is electron transport chain, you're going to see that's where you're going to generate most of the ATP, okay? And the way that the ATP is generated, just like I said, is similar to what we talked about in photosynthesis, all right? So again, super important diagram, okay? Because it provides an overview, tells you how many ATP are made at each one of these phases, right? And then also shows you where NADH and FADH2 are produced, okay? When we talk specifically about each phase, or when I talk specifically about each phase in the recorded lecture, there's then more detail there, okay? But I wanted to start off with you guys just understanding or just becoming a little bit familiar with these 4 phases. So we should know the 4 phases of, you know, complete glucose breakdown, or the 4 phases of cellular respiration. They are here: glycolysis, preparatory reaction, citric acid cycle, electron transport chain, okay? And like I said, the recorded lecture talks about each one of these phases in more detail. Okay, does anyone have any questions right now? So let me just show you, I wanted to just show you one of these videos just so you guys can see that they're helpful. Maybe louder.
Can you turn it up?
Yeah, I got 1 second. I gotta find the volume on my computer. Oh, my volume's turned all the way up. I don't know why it's doing that. What the hell just happened? 1 second. Where'd it go? That's annoying. 1 second. I don't know what the heck I just did. I got rid of my, Anywho, it's right here. And if the volume doesn't turn up, it's fine. I'll just sort of, you can watch any of these on your own, right? They're all in here. So let's just look at this glycolysis one. I think that's the one I was trying to look at. Oh, they're all here, duh.
Cells derive energy from the oxidation of nutrients such as glucose.
The volume's turned all the way up.
Oxidation of glucose to pyruvate occurs through a series of steps called glycolysis. The energy released during these oxidation reactions is used to form adenosine triphosphate, ATP, the energy currency of the cell, and NADH. During the initial step of glycolysis, energy from ATP is invested into the process. 2 phosphates from ATP are transferred to a glucose molecule. The result is a 6-carbon sugar diphosphate molecule and 2 low-energy adenosine diphosphate molecules, or ADP. This 6-carbon sugar diphosphate molecule is then split into 2 3-carbon molecules. During the energy harvesting steps of glycolysis, each 3-carbon molecule is converted through a series of steps to pyruvate. Electrons are transferred to the coenzyme NAD+ to form NADH, and ATP is produced. Under aerobic conditions, the pyruvate is further oxidized to yield more ATP, and under anaerobic conditions, the pyruvate is converted into lactic acid.
All right, so that was just an example of one of the animations that's there. It mentioned at the end about aerobic versus anaerobic. So what does it mean when we say aerobic? What does that mean?
That there's movement.
Nope. What does aerobic mean?
It uses air or oxygen.
Oxygen, yeah. So aerobic means when oxygen is present, or that it can utilize oxygen. Anaerobic, right? No oxygen is around. So if you heard about like fermentation, right? If you think about how you make beer and wine and other alcohol, right? It's fermentation, right? So there are, you know, yeast and other bacteria can go through fermentation, and they cannot utilize oxygen, okay? So I talk about that in the recorded lecture, and we'll talk about that a little bit more as well, okay? So glycolysis will happen no matter, or whether or not oxygen is around. So in both anaerobic and aerobic conditions, glycolysis will occur. And then if there is no oxygen around, there's sort of an alternate pathway that can occur, okay? But if there's oxygen around, then you'll move through those 4 phases that I showed you guys at the beginning of class, okay? Okay, so like I said, there's a lot of information in that chapter. You're gonna have to spend some time with that recorded lecture, okay guys? So I'm gonna leave the rest of that to you guys to watch that recorded lecture, and we'll talk more about this in class on Thursday. All right, does anyone have any questions right now? Again, remember all these animations are there, they're helpful, okay? So that's just another resource for you guys. Any questions about that right now? No? Okay, all right. So what I'm gonna have you guys do, okay, is this here? Yeah, okay. If you guys look at, where is it? Not there. It's right here. So I'm under this week's module, right? Module 8, October 12th. If you guys, everyone should be able to go onto Moodle, okay? Download this Lab Exam 1 review sheet, or just open it up as a Google Doc, okay? So I have to go pick up my son from school. This is the first time they're back in school. So I'm gonna randomly assign you guys to groups, okay? So it'll randomly put you in meeting rooms. At least one of you needs to open up this Lab Exam 1 review sheet as a Google Doc, right? So if I go, well, you guys should be able to do that. I don't need to, otherwise I gotta go, I don't have my email open, so I'm gonna have to go to my Google Drive. It'll take me a minute. But what I want you guys to do, again, download this Lab Exam 1 review sheet, okay, in your groups, and you're gonna start working on filling in that Lab Exam 1 review sheet, okay? You'll stay on the meeting, and when I come back, I'm gonna check in to see where you guys are at. So each group will write their names on the top, or whoever's recording the information, write your names, full names on the top of that Google Doc, and share it with me, okay? So that I can see how you guys are working on it. And then, like I said, once I come back, I'll check back in on everyone. But let me just first, just kind of go over some of what's on here quickly. Okay, so again, these are all the labs that we did. This is what your lab exam is on Thursday, right? So for, again, you're filling in details here. So for example, for these metric system conversions, maybe put just one example there or something like that. Or the units in between, meters to centimeters to millimeters, right? Add in the actual factors there, right? Are they, so from micrometers to millimeters, what's the factor that it differs by? I'm not really sure why I have it written this way. It's kind of weird the way I have it written. Anyway, you can fix that, right? You want to go from usually largest unit to smallest unit. So if you go from millimeters to micrometers, what number or what factor do they differ by? 100, 10, 1,000? 1,000. Good. So I would add in that information here, right? Remember, I kind of provided an extra document, I think, in the metric lab somewhere up there and in those weeks in Moodle that had that info in there for you, okay? So maybe add that in there. For temperature, you know, basic conversion from Fahrenheit to Celsius, add that in. It's in all of the lab information that you guys have, okay? So I want you guys to work on this together, to be filling this in using all of the information you have. Most of it's just already on Moodle, but the lab reports that you did, the labs that you filled out, whatever, you guys have all of this information, okay? There's a few definitions here. In terms of the parts of the microscope, don't worry about adding in anything in for the review sheet there, but any of this stuff here, okay? Add in any information or any detail that you learned from the labs. So you're basically sort of filling in this outline just like you guys do for the exam review sheet. Does anyone have a question in terms of what they're gonna be doing, you guys are gonna be doing for like the next probably, I don't know, 20 minutes or so, and I'll check back with you. Like I said, as soon as you open it as a Google Doc, share it with me so that I can see you guys working on it. Put all of the group members' names. Go ahead.
Is it the lab exam 1 review sheet that we have right now?
Yeah, lab practical 1 review sheet. Yeah, lab exam 1 review sheet. Yep. I'll show you where it is again. It's right here, right? Lab exam 1 review sheet, okay? Like I said, open it as a Google Doc, share it with me, and then you're gonna write your group members' names on it. I'm gonna divide you guys into groups, okay? All right, any other questions?
So only one person will have to do it?
Well, one
We'll
person
write
will
it?
be the one recording, correct. Only one
Okay.
of you has to record it. Then the rest of you, then just add everyone's names on the top of that Google Doc. Yep. Other questions, guys?
So when we submit, I just want to make sure when we
You're not submitting anything, you're sharing it with me so that I can check back to see where you guys are at. And so I'm basically going to be seeing what you guys are doing as you're doing it, because you're sharing the document with me. So you're going to open it as a Google Doc and you're just going to share it with me and share it with all your group members or whatever, right? It doesn't really matter. As long as you share it with me and you're all working on that one together, that's fine. Okay? Other questions? All right. And so I apologize if you have somebody in your group that's just not really here, but that's their problem. So you guys will find that out. So I'm going to randomly assign you guys to groups, okay? It'll just send you to the different breakout rooms. Like I said, work on this, and then I'll end up bringing you guys all back to the main room probably in like 20 minutes or so. Maybe, I don't know, 25 minutes, okay? And then if you have questions about this, I can answer those questions. But this is meant for you guys to review so that you're ready for that lab exam on Thursday. All right, one last chance for questions. All right, I'm gonna send you guys to rooms, probably like 2 or 3 people in each room, okay guys?
Excuse me, Professor. Excuse me, Professor.
Professor.
Yeah.
I was trying to share my screen with my my group, and it kicked me out and brought me back to the main meeting. And
All right. I'll just, 1 second.
I was in breakout room 8. If you could put me back in.
I can put you, who was that? Who said that?
Jason.
All right. There's nothing even on here. That's awesome. Still waiting for everyone to come back. All right, so as we wait for everyone to come back, if you still need to finish those, you guys can continue to work on those and just keep it as it's shared with me, and I'm just gonna check. Again, that's gonna be your attendance for today, so I'll just make sure that you guys completed it. So if you need a little extra time to complete it, that's fine. Once we finish here, you guys can kind of continue with that. But I wanna just see if anyone has any questions. Anything that you want, or anything you want me to go over a little bit more or clarify more? Anything here for like from the first lab for metric microscope?
I have a question.
Yeah, go ahead.
Give me a moment. Relating to the microscopy. If you don't mind, could you please re-explain parfocal again?
Yeah, so actually parfocal, that's probably, I don't know that I really went over that kind of explicitly when we were doing the lab. So I'm glad you asked that question. So you guys kind of, you know what it is, because when you actually use the microscope, this was always happening. So it's the idea that if something is in focus, say under low power, right? When you switch to high power, it will still pretty much be in focus. So that's what it means if the microscope is parfocal. So remember how we always say when you're using the microscope, you should start at scanning, get it into focus, and then switch up to the higher power. The reason why that works well is because the microscope is parfocal. Is that okay?
Thank you very much.
Yep, you got it.
And also, just to confirm, you said we do not need to do the what do they call it? The parts of a microscope.
Well, for this, you don't need to do the parts of the microscope, but you will need to know the parts of the microscope.
Okay.
For the lab exam. So there's definitely a question or two that has a diagram, a microscope diagram, that I asked you to identify the part. You should also know the function of those parts as well. Okay, so in fact, you need to know that, but I just told you guys, don't worry about like identifying parts there. If you want to kind of finish this, you could add in the function of some of those main parts, right? So that's basically, if we look, let me go back. That's all, it's in the PDF, I believe. So let me just pull that up just to kind of clarify that. Anything else you guys want me to clarify too as I pull this guy up real quick?
For laboratory 3, I wonder, like, for you say negative and positive, do you want me to explain like each one? And then for the other one for starch and stuff, do you want me to like break it down, like when we had the water and like potato juice? You want to break it down like that?
Well, I mean, it's more understanding what those tests are for. So like if I showed you a picture on the lab exam, say for iodine, right? And I showed you one tube where it was yellow or amber color, and the other tube was dark black or dark blue, and asked you which tube contained starch, what would your answer be? So it's more about identifying that. So, right, so one amber colored tube, one blue black colored tube. Which tube contains starch?
Gotcha now.
Yeah, right. So it's questions like that. So I'll kind of provide the scenario, right, or give you that information and say, tell me what had starch, you know, which one had starch. So you need to know for each test what indicates it's positive or that that molecule is present.
Okay, I got it.
Right?
Thank you.
Yep, you got it.
So like the change of color, is that what you're looking for? Like we have to write down in like the review, like what color it changes to?
You're not gonna have to write it down. You're gonna have to answer the multiple choice question, right? So if I show you a tube that's blue-black and I said I did an iodine test on this tube, does it contain starch or not? Yes or no? If it's blue-black, what's the answer?
Yes.
Yeah, so it's just understanding that and being able to answer questions like that, right? So knowing what, why is this not downloading this? Knowing what each test tests for, and then when you see a tube and you look at that color change, you need to be able to tell me whether it has starch or whether it has glucose or not. Does that make sense?
Yes.
Okay. Sorry, I'm just trying to pull up I don't know why it's not, so annoying. Keep asking questions if you guys have it. I'm just pulling up I can let that download as I have this guy up. Other questions you guys have? So what color for the burette test indicates that there's protein present? What color change? What color does it become? Starts off blue, becomes what? Purple.
Purple.
Good. What about for the Benedict's test? If there's sugar present in your sample, again, it starts off blue. That's the negative test. What indicates that there's sugar present?
Dark orange.
Brad, say.
Orange.
Yeah, like orange, greenish, orange. There's a couple different colors that could turn. Okay. So yeah, for that stuff, that's really what I mean. Now for controls, it's more of kind of what do you mean when we say something's a negative control versus a positive control, right? In every experiment that we did in that lab, there was a negative control. And in the lab report or in the questions under the table, it asks you about that, right? So what does it mean if something's a negative control, or what in general was a negative control in all of those experiments from that lab? Yeah, water, right? We know water doesn't contain any of these things, so it's our negative control. If you got a positive result from your water sample, then there's something wrong with your experiment, right? Or it's contaminated or something like that, right? So that's why you do that negative control. And then what about positive control? So say for the starch experiment, what would be the positive control in that experiment, or in an experiment like that? What would you use as a positive control if you were testing for the presence of starch? It's not a trick question. What do you think?
It would state if starch was present.
Yeah, but what would actually be your tube that you would consider your positive control? What would it have in it?
Glucose.
For starch. For starch.
Starch.
Like onions. No, it was
Starch.
Potato.
You would actually have starch in it, right?
Potato.
Yeah. No, starch, because
it was
starch,
the onion.
but that's not technically really a positive control. That's one of your test samples, right? The actual positive control in an experiment is something that you know should automatically be positive. Not necessarily ones that test positive, ones that you know beforehand, right? So if you had starch solution, you do that experiment and it doesn't turn blue-black, what might that indicate? Something's wrong with your iodine solution or something like that, right? Therefore, all the rest of your samples are going to be messed up too, right? So negative control is something that you know should be negative, like water. A positive control is something that you know should test positive for that. So if you're testing for starch, your positive control tube should have starch in it, right? If you're testing for glucose, your positive control should have glucose in it. Does that make sense to everyone? Okay. Other questions?
What do you want us to write for like the onion cell versus cheek cell?
That's more about like what they look like in terms of like the shape of them. Yeah.
Okay.
The appearance of them. Yeah. Just put a little description there. So for this lab, I just wanted to make sure, just remind you guys, right? Parts of the microscope, not the dissecting scope, but the compound light microscope, right? You guys labeled these in your lab report or whatever for the lab assignment. So you should know these main parts of the microscope and also the function of the main parts that are listed here, okay? Other questions, guys? No? We're all right? Oh, and also for the cell, right? Remember, you also had an assignment where you labeled parts of the cell, right, from diagrams. That also will be on the lab exam. So meaning I'll show you a picture of an animal cell or a plant cell and say what organelle is labeled at A or whatever. So you should be able to identify those organelles from a picture.
I have a question, Professor. Will we be able to use this review for the lab exam or not?
Yeah, you guys are taking it on your own. So yeah, that's a good idea, right? You guys all went through this review. The best thing that you probably could have in front of you is this review sheet completed, because it's quick reference, right? Because remember, you don't have a lot of time to complete these assessments And that's the reason why, right? If I gave you an endless amount of time, you can just read up and search everything and not really have to know it. So yeah, definitely have it with you. Other questions, guys? No? Okay, so like I said, make sure, I don't know if those broadcast messages I know kind of flash quickly. Make sure on your shared Google Doc, everyone's names are listed right at the top, whoever worked on it. If somebody was in your group and they weren't really there and they didn't help, their name should not go on the Google Doc, okay? So whoever worked on it with you, and I can see that, by the way, you guys, in the Google Doc, I could see who does what and who did what. So who worked with you, right? First and last name on the top, and then just make sure it's shared with me. I think most of you guys did that, but I think there's a couple that might be missing, okay? I will use that as attendance for today. Like I said, if you need more time to work on it, keep working on it, and it's shared with me. But I really just wanted you guys to do this so that it got you reviewing the material and you could ask questions. You're not gonna have to submit that as an assignment, right? It's part of your attendance for today. Okay, and obviously helps you study for the lab exam. I'll stay on if anyone has more questions, okay? Otherwise, I will see you guys. Remember, you don't have to log in for the lab meeting on Zoom, okay? Either on Tuesday or Thursday. I'll see you guys 12:30. Oh, on Thursday. There's another room change, and I apologize, but it's for the better. This was the room that I was trying to get in. It's our biology classroom. I'll put it in the announcement, but it's HH121. That's where class will be, okay? So we'll be there from now on. So on Thursday, it's in person if you choose, or obviously via Zoom for lecture, okay? At the end of lecture, like I said, around 1:30, you'll be taking that lab exam. And then when you're done with it, you can just log off and you're done for the day. Okay, guys? So Henderson Hall 121.
So on Thursday, we do not have lab at all, just to confirm.
Correct, there's no lab. We don't have any in-person labs for a while, right? Until November.
But we won't meet. But we won't be meeting on Zoom either.
Nope,
right?
we won't be meeting on Zoom either this week. That's correct. All right. So like I said, I'll stay on if you guys have any other individual questions. I'm here, but otherwise I'll see you guys on Thursday at 12:30. Okay.
See you on Thursday.
Bye.
Thank you. Bye.
Right, bye.